success. In my presentation, I will try to illustrate the stages of our evolutionary path in the study application of genetic control of pests and vectors by showing to you the key points of this path. Let's start with a classic definition of genetic control shared by the International Atomic Energy Agency and the World Health Organization. Genetically based methods of vector control focus on this dissemination of genetic traits designed to reduce a vector capacities to reproduce or to spread human disease. As you can see, the known strategies are clustered into two main groups: self-limiting and self-sustaining, depending on whether the effect stop or persist once the release of sterile males or modified females stop. In this presentation, we will essentially deal with the self-limiting strategies and the improvement contribution given by the use of the Wolbachia bacterium. The skills of our research group are rooted in the study application of the classic SAT, well described before by Romeo, the mother of all the control strategy that relies on the release of large amount of radiation sterilized males to induce high levels of sterility in the larger population until eradication of the pest of the vector when it is possible. As you can see, ENEA was involved in a SAT campaign. It was the first in Europe to control the mayfly. Although many years have passed, this experience has taught us a lot about the complexity of SAT program. One of the most exciting aspects is that although all the control strategies are imperfect and therefore can be improved, can be integrated, innovated by each according to own skills to reach the most sustainable cost-benefit ratio. In this slide, you can see the innovative contribution of molecular biology, but even before there had been the observation in nature of unexplained form of sterility, reciprocal sterility between different Culex populations. Thanks to an increasingly holobiontic vision of living beings, in last decades, interest in the study of a functional symbiotic microorganism has grown. Among the most studied bacteria, we find Wolbachia, because we know it's a real manipulator of the reproduction of its host, including the capacity of inducing different patterns of conditional sterility. Our interest in the genetic control of the Asian tiger mosquito has been gradually increasing during the 20 years collaboration with the Centro Agricoltura Ambiente led by Romeo Bellini, and participated to projects coordinated by the International Atomic Energy Agency with the aim to evaluate the potential exploitation of Wolbachia in the framework of an enhanced SAT strategy. Thanks to this experience, we have learned to value more and more the function of Wolbachia and its biological properties. Just a short introduction to Wolbachia for those that are unfamiliar with this endosymbiontic bacterium. It is not easy to have a precise estimate of the incidence of infection in nature, but it is estimated that more than 50% of insects in nature live in symbiosis with this bacterium. We are talking about an alpha proteobacterium. It was first reported in Culex pipiens gonads with no pathological symptoms. It is very widespread in arthropods, infected gonads, and secondarily somatic tissue of the host. It's transmitted predominantly through female germline, and as I told before, the infection is associated with the manipulation effect on host reproductive biology. For those who work with the sterile male-based methods, the most important phenotype induced by Wolbachia is the cytoplasmic incompatibility. It is a conditional sterility, not absolute sterility, occurring when in a population we have infected and uninfected females that mate, or males and females harbor different Wolbachia cytotype. This perfect mechanism of sterility is due to an abortive development of a dysfunctional chromatin condensation events. The incompatible insect technique can be considered today an evolutionary branch of the SIT. It relies on the exploitation of a bacterium instead of radiation. However, in order to develop an IIT against mosquito or other species, we need to handle a strain that produces male reproductively incompatible with natural females. For what it concerns Aedes albopictus, no incompatible strain have been found in nature. In fact, all individual sampled are harbor two Wolbachia strain, type A and type B. For this reason, in 2009, we generated an artificial lab line replacing the natural infection in Aedes albopictus with a Wolbachia taken from Culex molestus living in Italy, generating a bidirectional.
Hello.
Excuse me. Okay.
Yeah.
Generating a bidirectional pattern of cytoplasmic incompatibility. In fact, when we are dealing with cytoplasmic incompatibility, we cannot generalize. In fact, cytoplasmic incompatibility may be unidirectional or bidirectional, depending on the bacterial cytotype that coexists in a population. In case of a unidirectional CI, we have just one level of sterility. In fact, one of the two infection type, in this case, the triple infection in comparison with the double natural infection, benefits of a reproductive advantage because this female may be successfully fertilized not only by males harboring the same Wolbachia strain, but also from the other one. Differently, when we have a bidirectional pattern of a CI, of a cytoplasmic incompatibility, the population are reciprocally sterile. We have two levels of sterility working. And this is the reason, because we have generated a bidirectional strain for an incompatible male-based approach. In this slide, I briefly report what are the research topics on which we have focused to enhance the upper line. First of all, we studied the incompatible male efficiency, monitoring and traceability of incompatible males in the environment, production and management of incompatible males, the possibility of combining cytoplasmic incompatibility and pathogen inhibition, the very important role of the model simulation, and the community accepted engagement in this kind of project. Since we are dealing with a control, just a moment, please. Since we are dealing with a control approach based on male sterility, the first research topic we focused on was the study of the sexual competitiveness of incompatible males in relation to wild males. This study were initially conducted in the laboratory and then in semi-field, in open field, in semi-field in collaboration with the Centro Agricoltura Ambiente. And more recently, in open field, I'm referring to the presentation of yesterday of Giulia. As you can see, results were very promising from the first level of experiment, where we found competitiveness indices not dissimilar between incompetent and normal males, regardless of the age of the tested males. The subsequent test in the semi-field reinforced results of previous experiment. Furthermore, in the semi-field experiment, we found a good level of competitiveness for 30 degrees irradiated males and excellent values of the capacity of induced sterility for incompatible males, as you can see in this male competitiveness trial. This difference could be not only a matter of mating competitiveness, it could be also due to the bigger size that incompetent males preserve during mass rearing, and to the fact that the cytoplasmic incompatibility ensures a 100% of sterility, while we know that irradiation presents a low level of residual fertility. With this premise, very encouraging premise, we have faced the field test in collaboration with the University of La Sapienza, essentially to evaluate the ability to induce sterility of incompetent males in wild females, starting from rather low release. This experiment were described yesterday by Julia, and I would not like to go into. Anyway, I just want to draw your attention to the fact that no control treatment were carried out to reduce mosquito population or to remove a breeding size during the trial. We want to evaluate the net effect of the compatible males. The observed induced sterility shown yesterday in 2018 and 2019 was achieved with the compatible to fertility ratio very low, well below the recommended 1:5. Obviously, we did not verify the level of suppression, but however, the induction of sterility, a careful study encouraged our ambition to think an ARP IRT based as an additional innovative tool in the framework of the integrated vector management in an urbanized area. Another very important aspect is that the quality and the accuracy of the field data were possible thanks to the choice of an appropriate experiment protocol shared with the University of Rome that allow us to evaluate the net contribution of the male release. The possibility of having, this is very important, a stable molecular marking for incompatible males made possible to have an accurate estimate on very important information, like the incompatible male survival, incompatible male dispersal capacity, IIT working efficiency, and also the level of eventual WPEP establishment after the end of the trial. All of these parameters are still research in progress, obviously. We are learning that SIT or IIT are big challenges when we move from a small to large scale due to the current technological limitation of these approaches. I often stated in my presentation anyway that the application of Wolbachia should not be seen only as a way of replacing irradiation with the bacteria. It would be reductive. We have to think something even with a bit of imagination and positive predisposition, what could be innovative solution that could shorten the production chain of males and reduce the workload of distribution. I shared with you this hypothesis, why, since IIT does not require irradiation facilities, DX of incompetent live could be produced in a centralized incompetent mosquito farm and then delivered to a local small facility, while incompatible males could be reared for release after the pupal sex. On the other hand, WP Wolbachia increases the sex efficiency. This has been shown from our group and also from the agency, the International Atomic Agency, due to a faster development of a male imagery. Advanced machine for mechanical sorting may increase further the efficiency of the sexy step. Studies are in progress. Another very important property of Wolbachia, and we know that some Wolbachia strains are well known to interfere with virus transmission, in particular the W male taken from Drosophila melanogaster. In 2007, we generated a new mosquito strain named ARP-M that basically restore a double infection in Aedes albopictus. One strain of the B supergroup and another strain of the A supergroup. We proved that this line to be incompatible thanks to the action of the strong action of the WP strain, and at the same time its vector was significantly reduced. After 3 years from its generation, this line is currently under careful evaluation for the tendency of the WB male to lower its density when exposed to high temperature, differently from W strain, WP strain that is very, very, very stable. Given the difficulty to carry out a complex experiment, mathematical models may support us to pursue long-term effectiveness and sustainability. Small-scale validation of the first model suggests that low immigration rates of wild females in the IIT-treated area can avoid the establishment of WP also in presence of a low rate of unwanted females released. Basically, a bidirectional conflict arises between two mosquito strains. Once the releases of the incompatible males cease, the wild strain will regain the upper end. The scenario treated in the second model is much more complex because it is an IIT application on a unidirectional CI basis against the Egypti. In this case, different male release strategies are under mathematical and validation evaluation, and some appear to prevent the establishment of the invasive cytotype. Basically, models help us in optimizing IIT, taking into account the difference between a bidirectional and unidirectional system. In this slide, I will just show you what we are evaluating on semi-field. When we have a bidirectional system, we can have really a local and transient population replacement. But if we are working in a not isolated area, we have in the context of a meta-population, local and transient population replacement may occur, in particular associated with a very high level of suppression, but it is a temporary unstable state. I'm going to the conclusion. Another very important aspect is I cannot leave out of the most engaging aspect, namely introducing the Wolbachia method to people, institutions, students, and also meeting potential investors. In Italy, we mostly experience great interest and desire to be part of the trial and the project. In Singapore, they published their experience, confirming our impression. In conclusion, I want just to summarize the most important point. Wolbachia-induced sterility does not burden male fitness in any case because males are only conditionally sterile and not absolutely sterile. This evidence enables lower release to be effective. Sterile male production and management is potentially easier, as I explained before, with the delivery over X. Incompatible males are traceable in the environment by the identification of the facial time. This possibility allows to easily monitor the frequency of the facial type to directly measure the effect of the control program and adjust the setup of the release. Cytoplasmic incompatibility, a pathogen inhibition can be combined in a single mosquito, increases the level of biosafety. Model simulation support the efficiency and safety of an IIT program, in particular when based on a bidirectional pattern. Positive sentiment of engaged community encourages IIT development. I thank you for attention, and I'm available for your question.
Thank you very much, Maurizio, for this very